Hello, and welcome to the Chapter 18, Parts 1 and 2 lecture on the control of gene expression. You should use the information in this lecture to complete the Chapter 18, Parts 1 and 2 guided notes, which of course you should complete before you attend class. All living cells inherit a set of genes from their parent or parents. These genes represent a set of instructions for making molecules such as RNAs and proteins. And these molecules are important because they perform all the little jobs of the cell. So cells maintain themselves and stay alive using mRNAs and proteins to do the work. The process of using the information in a gene to make one of these products is called gene expression, and this process has to be carefully controlled. Gene expression involves both transcription and translation, which we discussed in the last chapter. I want you to imagine an orchestra. Now imagine that all of the musicians in that orchestra are all playing their instruments all at the same time and just doing whatever they want. It would sound awful, and it certainly wouldn't sound like music. No, instead, in order for an orchestra to make beautiful, intricate music, it has to coordinate its efforts. Only specific musicians get to play their instruments at specific times. The same sort of thing is true for cells. In this analogy, the orchestra represents the genome of a cell, or the entire collection of genes inside that cell. In order for the cell to stay alive and to stay organized, only certain genes can be active at certain times. The cell has to decide which ones need to be turned on and which genes need to be turned off at any given moment. So even though all cells inherit a set of DNA from their parents, none of them use all of their DNA all of the time. Having the ability to control the pattern of gene expression is really important to cells for a couple of reasons. First, it allows cells to respond to a changing environment. Let's consider the bacteria, the many species of bacteria that live inside your guts. Now, every time you eat something, you're not only feeding yourself, you're also feeding your gut bacteria. Now let's consider what might happen if you eat a brand new food that you've never had before. It's really different than your usual diet. That new chemical may be toxic to some of your bacteria. So some of the bacteria in your guts may have to activate genes that help them protect themselves from that new chemical. Other bacteria may love this new chemical. They may want to feed on it. So they have to activate genes for enzymes that they don't normally use so that they can break down that food source and use it as their own as well. In this way, different kinds of bacteria can use different sets of genes to respond to this new environmental factor. We all started out life as a fertilized egg, and at a certain point, that fertilized egg divided into a little ball of cells. Now, at that time, the cells in that little ball were essentially all the same. They weren't very different from one another. But at a certain point, those cells needed to differentiate. They needed to begin to change into different kinds of cells so that that embryo could develop organs and organ systems. Now, think about this. You have over 200 different types of cells inside your body. They differ in their structure and their function, and some of them are really wildly different from one another. However, if we were to examine the DNA inside those cells, we'd find that the DNA is identical. The DNA inside a muscle cell is exactly the same as inside a bone cell or a nerve cell. How can this be? Well, again, it has to do with the control of gene expression. At a certain point in development, these little stem cells inside that little embryo are going to begin to turn certain genes on and other genes off, so that muscle cells begin to develop that only use muscle genes, and red blood cells develop that only use red blood cell genes. By turning certain genes on and off in certain patterns, cells can differentiate. They can become different types of cells. The cell can turn off gene expression at many points along the pathway. The most common method of control occurs at the level of transcription. The cell can affect the DNA in such a way that it can't be transcribed or copied into mRNA. And if it can't be transcribed, it can't be translated. So that gene has essentially been turned off. Cells can also encourage or speed up the rate of transcription of a particular gene, thus making more transcripts. The more transcripts that are made, the more of that particular protein can be made at one time. Cells can also encourage or discourage the making of certain protein products after transcription has occurred. They do this by either affecting the process of mRNA processing or by affecting the process of translation. This method of control isn't as common as transcriptional control, but it's still very useful to the cell. We refer to it as post-transcriptional control. In this lecture, we're going to focus on transcriptional control because it's by far the most common method. Now, to understand how cells turn transcription on and off, you need to understand a little bit more about DNA, what it's for, and how it's put together. So far this semester, we've learned that DNA consists of genes, and genes are these recipes for making molecules like proteins. Now, while that's true, it turns out that genes are actually a very small percentage of the total DNA. That between the genes, there are these long sections of DNA that don't code for anything. Scientists used to call this DNA junk DNA when they didn't really understand what it does. And now we have more of a clue about what it's for. So let's explore that in more detail. So as we've discussed before, DNA, of course, contains genes. Genes are the recipes for making such molecules as proteins and RNA. They are the instructions for making things like enzymes, channels, receptors, and many other important cellular tools. There's one other category of proteins we haven't discussed yet. These are called regulatory proteins. Regulatory proteins are kind of interesting. These proteins will bind to the DNA and they turn the transcription of other genes on and off. Now, there are genes built into the DNA for these proteins, and the genes that make regulatory proteins are called regulatory genes or sequences. So within the DNA, in amongst the genes for building things like enzymes and channels and receptors, are genes for making proteins that will control the expression of those enzymes, channels, and receptors. There are many different types of proteins and other molecules that can bind to the DNA and turn it on or off, these so-called regulatory proteins or molecules. Activators have a tendency to turn on transcription, to encourage transcription, whereas repressors have a tendency to turn it off. Co-repressors are little molecules that help the repressors, so they also help turn a gene off. Things we call inducers can sometimes turn genes on and sometimes turn them off, depending on what other molecules are involved. We'll look at an example of an inducer later on. And finally, in eukaryotic cells, we have these molecules called transcription factors. You might remember them from chapter 17. These are molecules that assemble on the promoter along with RNA polymerase and help transcription begin. We'll see it in part 3 of the chapter. Next, let's discuss the so-called junk DNA. Many sections of junk DNA consist of these things called control elements. Now, control elements are just stretches of DNA. They're made of A's, T's, C's, and G's in a particular order, just like the genes are. However, control elements don't code for anything. They are not the instructions for making RNAs or proteins. Instead, these sections provide areas for other machinery to grab onto the DNA and manipulate it in certain ways. For instance, in the last chapter, we learned about a little section called a promoter. The promoter provides an area for that enzyme, RNA polymerase, to grab onto the DNA and begin transcription. There are several different important types of control elements, including operators and enhancers and many others, and we'll discuss them as we get to them. Here are the three types of control elements you should be aware of. First, we have the good old promoter. To remind you, the promoter is a section of DNA that's right next to the gene, and it provides an attachment point for RNA polymerase. Once RNA polymerase has attached, it can begin transcription. In prokaryotic cells, there's a section of the promoter called the operator. Here I've shown the operator as a big, fun, red, candy-like button. And I've shown it as a big red button because this operator can serve as a switch for the gene. Depending on what attaches to the operator, it can either activate transcription or it can discourage transcription. Finally, we have sections called enhancers. Enhancers are referred to as a distal element because they're actually pretty far away from the actual gene. They're way over here. Enhancers only occur in eukaryotic cells, and when certain transcription factors grab onto them, it will allow them to interact with the promoter and turn on that gene and start transcription. Now, how do they do that, being so far away from that promoter? Well, we'll see that in part 3 of the chapter. Now, I need to point out that this little diagram is a little bit deceptive, and that's because operators occur in prokaryotic cells, whereas enhancers only occur in eukaryotic cells. So you won't ever actually see a piece of DNA like this that has both operators and enhancers in it. Let's put this all together. Regulatory molecules, such as RNA polymerase and things like activators and repressors, bind to control elements in the DNA, and they either encourage the transcription of a gene or discourage the transcription of the gene. By doing so, they control the amount of that protein or RNA product that is made from that particular gene. The binding of regulatory molecules to control elements can also control the speed with which certain genes are transcribed and used. For instance, here we have 3 different kinds of cells. We've got brain cells, liver cells, and skin cells. Within the DNA of those cells, there's this particular gene for a particular protein product. Now, since all of these cells have exactly the same DNA, they are going to carry the exact same copy of that gene, and they're going to contain the same control elements for that gene. However, there are different molecules that can bind to these control elements, and that can speed up or slow down transcription. For instance, whatever this protein is not needed very much in the brain. So certain molecules bind to control elements and it slows it down. So it makes that product but very slowly. More of that protein is needed in the skin, so different control elements bind to it, and that makes it transcribe at moderate levels, at kind of medium levels. And finally, this protein is needed a whole lot in the liver, so a different regulatory molecule binds to those control elements, and it gives you a high level of transcription. So the use of these regulatory molecules really gives the cell a lot of control over which proteins it makes, how much it makes, and when it makes them.